My name is Chibowaye, and on behalf of my co-authors, Abdullah Feroz and Eric Holland, I will present some basic molecular biology techniques, as well as advances in these techniques that have contributed to the pathologic diagnosis and discovery of alterations and targets in gliomas. The past 3 decades have witnessed unprecedented progress in the development of groundbreaking techniques in molecular and cellular genetics that enable the study of DNA from both normal and cancer cells. During this presentation, we discuss how clinically relevant genomic alterations are now being incorporated into the 2016 World Health Organization or WHO classification of tumors of the central nervous system. We then discuss some of the molecular biology-based techniques that have led to the discovery of mutations which are now part of the WHO update. These include some of the earliest techniques in isolating, amplifying, and studying DNA changes associated with cancer development, such as polymerase chain reaction, or PCR, and fluorescent in situ hybridization, or FISH. We also discuss advances in these approaches, including complex, large-scale techniques such as whole exome sequencing, comparative genomic hybridization, and RNA sequencing as a way to quantify messenger RNA expression from DNA. We then proceed to techniques used to detect epigenomic alterations, which are alterations that affect gene expression without changes to the DNA sequence, including mutations to histones, which are unique to midline diffuse gliomas, as well as MGMT promoter hypermethylation, which dictates susceptibility to temozolomide chemotherapy. Finally, we discuss how animal models are used to determine the relevance of mutations during tumor initiation, progression, and response to treatment. The World Health Organization classification of tumors of the central nervous system was updated in 2016 to incorporate advances in the molecular profiling of tumors within the central nervous system. The most recent iteration includes both genomic and epigenomic alterations that define specific subtypes of adult and pediatric brain tumors with significant clinical consequences for therapy and prognosis. For adult gliomas, the most significant updates include the recognition of IDH1 wild-type versus IDH mutant tumors as distinct entities within diffuse astrocytomas, glioblastomas, and oligodendrogliomas. In pediatric gliomas, there is now a distinct H3K27 mutant diffuse midline glioma, a marker that indicates poor prognosis. These distinctions are allowed for improved stratification of patients who, for instance, have IDH1 wild-type tumors associated with an aggressive phenotype that may require closer surveillance. We anticipate that future iterations of the WHO CNS tumor classification will incorporate more genomic alterations that will not only aid in diagnosis and surveillance, but also help guide targeted treatments specific for those genomic alterations. Next, let's discuss some of the techniques that have contributed to this advance in diagnosis. The first step in characterizing aberrant tumor DNA is the ability to amplify small amounts of DNA found in tumor cells without introducing new errors, such that enough DNA substrate material is generated for further molecular cytogenetic studies. Polymerase chain reaction, or PCR, allows the large-scale replication of DNA for additional study. After amplification of target DNA sequences, the simplest next step is DNA sequencing, allowing a readout of the genetic code and future comparison among sequences obtained from diseased host cells and normal controls. In this way, candidate mutations resulting in more important changes in cell behavior have been identified. PCR has revolutionized the applicability of gene-based technologies to clinical medicine. It permits the identification and amplification of DNA sequences from any source, as long as part of that sequence is known. Therefore, any known DNA sequence can be detected in any sample containing genetic material, including tumors. Initially, 2 oligonucleotide primers, or short pieces of synthetic DNA with sequences designed to anneal or stick to regions flanking the DNA sequences to be amplified, are designed and synthesized chemically. DNA containing the target sequence to be amplified is mixed together with oligonucleotide primers, free nucleotides, and heat-stable DNA polymerase enzymes. Each cycle of PCR requires heating the mixture to melt double-stranded DNA into two single strands, cooling to anneal primers to the template DNA to be amplified, synthesis of a new complementary strand from free nucleotides, and finally, melting and annealing to separate new from old strands of DNA. Each cycle results in doubling of the initial template quantity. Next, the PCR product is separated on a gel, and DNA fragments of expected size are purified and collected for downstream application, including sequencing. These results are then aligned to the human genome to determine where mutations exist, for instance. PCR can also be used to detect or quantify particular DNA sequences in a sample. For example, to detect the presence of viral sequences or to determine whether a tumor sample contains known sequences or a known genetic alteration. PCR products can also be used to generate purified DNA of known sequences for use in the construction of novel DNA molecules. Perhaps most important, PCR is now used routinely to detect new mutations in amplified or deleted regions of tumor DNA and new mutations in known or putative tumor suppressor genes and oncogenes. After isolating and amplifying target DNA sequences, researchers may then introduce or remove specific DNA sequences in cells and animals to understand the function of these sequences. PCR has undergone tremendous advances since its conception in the early 1970s. Initially, and as late as the early 1990s, researchers used two-dimensional chromatography and radiolabeled nucleotides to sequence PCR-amplified short strands of DNA. Since the advent of fluorescence-based techniques, DNA sequencing has become automated, allowing simpler, faster, and more accurate sequencing of significantly larger stretches of DNA. One of such approaches is called whole exome sequencing. The human exome represents less than 2% of the human genome, but contains approximately 85% of known disease-causing variants. Sequencing the exome only is said to be a cost-effective alternative to sequencing the whole genome because it bypasses the need to sequence introns. First, DNA is sheared and then ligated to unique adapters that enable sequencing on specific platforms. This pool is then amplified by PCR and hybridized to fluorescent oligonucleotides specific for exons. Following removal of unhybridized DNA, the genomic DNA with fluorescent oligonucleotide hybrid should now be enriched for exons. A sequencer is then used to detect each nucleotide attached to the fluorescent probe and cross-referenced to the human genome to identify mutations. Cancer, especially in its most malignant forms, progresses by accumulating multiple errors in the genomes of affected cells. Many of the genetic errors result in malfunction of the DNA proofreading mechanisms, which normally function to repair damaged DNA. As a result, the entire genome of an affected cell becomes prone to error, resulting in global genomic instability. A consequence of genomic instability is susceptibility to single and double-stranded DNA breaks. At the chromosomal level, these breaks can result in very large losses, gains, or translocations of chromosomal regions. These chromosomal areas encode genes or regulatory elements of genes involved in cancer initiation and progression. An identification of these abnormal chromosomal regions is important for a general understanding of cancer biology. As a consequence, cytogenetics, which encompasses the study of chromosomal structure, has become a central part of cancer biology. The prototypical molecular cytogenetic technique, fluorescence in situ hybridization, or FISH, is currently widely used for the identification of chromosomal aberrations in cancer. FISH can readily identify large chromosomal aberrations, such as rearrangements and deletions. FISH requires the use of a synthetic oligonucleotide probe containing fluorophores that correspond to a known region of the genome. The probe is then hybridized to the target DNA, usually genomic DNA of interest. After synthesis of the probe, both the probe and the target DNA are denatured and incubated to allow annealing of the probe to the target DNA. These probes, modified with fluorophores, can be visualized directly because of the fluorescence emitted by the probes. Structurally normal target DNA will exhibit a signal at the expected site of the chromosome, whereas aberrant signal will be seen in translocated DNA, such as 1p 19q code deletion, which is required for the diagnosis of oligodendrogliomas. Changes in signal intensity and location will be seen in amplification, while no signal at all will be seen in deletion. Although FISH has helped to identify many chromosomal abnormalities associated with non-solid tumors, this technique is imperfect for solid tumors. In advanced solid tumors, it is often impossible to interpret tumor chromosome spreads with accuracy because chromosomes are often altered beyond recognition in solid tumors. Comparative genomic hybridization, or CGH, represents an advance in FISH. For CGH, DNA is extracted from a test sample or tumor cells. The test or tumor DNA is then labeled with a fluorescent dye of a specific color, while DNA from a normal control or reference sample is labeled with a dye of a different color. The two genomic DNA, test and reference, are then mixed together, hybridized to specific sequences on the microarray spread. Because the DNAs have been denatured, they are single strands. Therefore, when applied to the slide, they attempt to hybridize with the arrayed single-strand probes. Next, digital imaging systems are used to capture and quantify the relative fluorescence intensities of the labeled DNA probes that have been hybridized to each target on the microarray. The fluorescence ratio of the test and reference hybridization signals is determined at different positions along the genome, and it provides information on the relative copy number of sequences in the test genome as compared to the normal genome. In particular, this technique is especially powerful for identification of changes in DNA copy number, such as increases in gene copy number that occur at the epidermal growth factor receptor locus, or EGFR locus, in a significant number of glioblastoma, based on higher levels of gene signal in the uniquely labeled tumor sample relative to control samples. To assess the consequence of genomic alterations for gene expression, we measure mRNA levels. Prior techniques have used microarrays, which utilizes a hybridization technique to detect and quantify specific mRNA transcripts. Advances in sequencing techniques now allow for more sensitive detection of mRNA transcripts without the need for hybridization. RNA sequencing uses the next-generation sequencing techniques mentioned previously for DNA to provide a glimpse of the relative predominance of RNA at specific moments in time. Here, the first step following RNA isolation is separation of coding from non-coding RNA. This is accomplished by taking advantage of the fact that coding RNA possesses a poly-A tail. Hence, poly-T oligonucleotide bound to beads can be used to selectively separate coding from non-coding RNA. This technique is particularly useful for the detection of fusion gene products linked to specific cancers, post-transcriptional alterations, and single nucleotide variations. Next, the RNA is transcribed to coding DNA, or cDNA, by a step known as reverse transcription. This is done prior to sequencing. To minimize errors generated during cDNA synthesis, some researchers have developed platforms that allow for direct RNA sequencing. This can be accomplished de novo without a genomic reference or with a genomic reference as guide. Once the cDNA is identified, it is quantified and cross-referenced to the human genome to identify specific genes associated with the cDNA. This is then quantified to provide relative gene expression of cDNA1 corresponding to gene X versus cDNA2 corresponding to gene Y, as shown here. Given the significant intratumoral heterogeneity within glioblastomas, capturing the diversity at the individual cellular level is imperative for finding therapies that would eradicate tumor-initiating clones within the tumor and determining how tumor responds to treatments at the clonal level. This need has resulted in the development of single-cell RNA sequencing, a variation of the traditional RNA sequencing technique. The main difference between both techniques is that in single-cell RNA sequencing, RNA is isolated from individual cells instead of a combination of cells, as in traditional RNA sequencing. For single-cell RNA sequencing, fresh isolated or fresh frozen bulk tumors are dissociated into individual cells. Non-tumor cells are sequentially removed from the heterogeneous mixture. Red blood cells are lysed and centrifuged out of the mix. Immune cells are depleted by fluorescence-activated cell sorting, or FACS, which utilizes the CD45 immune cell surface marker to sort and select CD45 positive immune cells out of the mix, leaving non-CD45 expressing normal and tumor cells. RNA is then isolated from individual tumor cells following sorting by flow cytometry. Ultimately, this technique will hopefully pave the way for discovery of clonal populations of tumor cells that drive tumor recurrence following treatments, such as cancer stem cells. This will allow for selective targeting of this clone to prevent tumor recurrence. DNA mutations are not the only form of alterations that affect gene expression. Unlike DNA mutations, epigenomic alterations do not alter the DNA sequence, but influence gene expression at different levels, such as transcription and translation. Examples of epigenomic alterations are aberrant histone modifications or DNA methylation. To assess histone modifications at the whole genome level, antibodies to the specific histone modification of interest are incubated with formaldehyde cross-linked DNA samples. The cross-linking procedure is performed to maintain proteins that are bound to the DNA in the bound state. The antibodies with histones and associated DNA are pulled down from the mixture. Next, the antibodies, histones, and DNA are denatured and dissociated, with the DNA eluted from the mixture given that this sequence represents the target. These individual DNA sequences are then sequenced, and the reads are cross-referenced to human DNA sequence databases. The genes corresponding to these DNA sequences are therefore believed to be associated with the histone targeted in the initial steps of the protocol. Ultimately, when this procedure is performed across multiple tumor subtypes and samples, unique histone modification signatures can be associated with expression status of genes. For instance, the unique histone protein H3K27 trimethylated is known to be a repressive modification, as it is associated with repression of promoter activity. Therapeutically, it is proposed that targeting these histone modifications, either by enhancing the pro-apoptotic associated marks or by inhibiting modifications that inhibit tumor suppressor genes, may slow progression of cancer. In glioblastoma, several agents targeting the repressive H3K27 ME3 modification are currently in development. 3-deazoxyniplanosin, or DZNP, has been shown to block the H3K27 ME3 or trimethylation mark and inhibit proliferation of GBM cells in vitro. Although DZNP is still in preclinical phase of trials, its discovery suggests that targeting glioblastoma with histone modification inhibitors may represent another therapeutic avenue for suppressing tumor progression. Another form of epigenomic alteration that affects gene expression are reversible chemical modifications to DNA nucleotides such as methylation. Promoter hypermethylation has been demonstrated to inhibit the expression of tumor suppressor genes. These epigenetic phenomena are particularly important for genes without sequence mutations. Expression of some DNA repair genes, such as O6-methylguanine DNA methyltransferase, or MGMT, has been shown to be suppressed via promoter hypermethylation. This epigenetic alteration was further demonstrated to confer susceptibility of glioblastoma to temozolomide. Altogether, DNA methylation is a strong epigenetic modulator of gene expression and therapeutic response in glioblastoma. Several techniques exist for determining whether a specific sequence corresponding to a gene's regulatory region, such as a promoter or enhancer, is methylated. Although bisulfite sequencing is the traditional approach for methylation profiling hundreds of genes becomes a tedious and painstaking process. The recent advent of whole genome methylation profiling, however, has made it possible to determine the methylation profile of hundreds of genes across multiple tumor samples in a short time period. To determine the methylation status of promoters, a bisulfite conversion is done to the whole DNA sample to be analyzed. This causes unmethylated cytosine to be converted to uracil, whereas methylated cytosine remains a cytosine. Following this step, the DNA is amplified with DNA polymerase, and the products are enzymatically fragmented. The fragmented DNA is applied to a chip that contains unmethylated DNA-specific oligonucleotides labeled with the specific fluorophore type, depicted in green, while another oligonucleotide targeting methylated DNA is labeled with a different fluorophore, depicted as red. Following hybridization and single base extension, the relative proportion of methylated to unmethylated DNA is calculated based on the fluorescence intensity of each fluorophore. This technique is used for the detection of MGMT promoter hypermethylation in glioblastoma. Following in vitro studies, cancer research seeks out in vivo relevance of the findings in vitro. It is speculated that in vivo or preclinical data better represents a human disease process because they're not influenced by in vitro artifacts, such as variability in media concentrations, oxygen availability, and passage number. Over the last few decades, several genetically engineered mouse models of disease have been used to study glioma initiation, progression, and response to therapy. One example of a genetically engineered mouse model that has yielded insights into the relevance of specific genes in gliomas are knockout models, in which genes are mutated or disrupted. Studies with knockout mice are meant to corroborate short hairpin RNA or small interferon RNA studies in vitro. Given that these inhibitory in vitro studies may be influenced by a number of factors affecting efficiency of knockdown, a knockout mouse model provides a more efficient way to determine the impact or complete loss of function or deletion of a gene during disease initiation and progression. Several strategies have been developed to induce the deletion of specific genes, such as conditional knockouts, where the DNA sequence vector that recombines with native DNA bears specific sequences at its flanks that can be recognized by DNA cleaving enzymes such as Cre-recombinase. Cre-recombinase can be introduced into the system by crossing a cell-type specific promoter-driven Cre-recombinase strain with a LOXP flanked gene of interest strain. In the progeny of these mice, bearing constitutively active Cre-recombinase, the gene of interest will be deleted as Cre-recombinase targets and cleaves DNA at the LOXP sites. Furthermore, the activity of the Cre-recombinase can be temporarily controlled by using a Cre-recombinase that can be regulated by exogenous drugs. Cre-ERT is a Cre-recombinase that is only active in the presence of tamoxifen. Therefore, the timing and impact of gene deletion can be controlled by giving tamoxifen at a specific time period of tumor initiation and progression. This approach is critical for determining the impact of specific genes on tumor progression in a complex environment. A reporter can also be used to track cells that have undergone recombination or have lost the target gene. One example is the luciferase reporter mice. Here, we show how luciferase activity tracks intracranial glioblastoma tumor cells that underwent Cre-recombinase-based deletion of P10, a tumor suppressor gene. Some genetically engineered mouse models utilize patient-derived cells to generate tumors in vivo. These are called patient-derived xenografts, or PDX. Although this technique allows for personalized studies of specific patient tumors, one disadvantage is the problems associated with the generation of a human tumor in a mouse model. This precludes the use of certain therapeutic agents such as immunotherapy, given that the host mice used for PDXs are immunodeficient or immunocompromised. The RCAS-TVA model has been used to recreate the genetic alterations found in human gliomas, resulting in the formation of mouse tumors that mimic the histology of human glioblastoma. Here, the replication-competent avian leukemia virus, family splice acceptor, or RCAS, vectors are designed to express the gene of interest. The virus bearing these constructs then targets cells with cell-type specific expression of the ALV type A receptor, or TVA. Ultimately, when the virus is delivered broadly, it only infects cells expressing the TVA receptor, which if engineered to be expressed by the nestin or glial fibrillary acidic protein promoters, will be specific for CNS stem or progenitor cells, or astrocytes. The transfer of some genes, such as PDGF, or combined oncogenic RAS and AKT, to stem or progenitor cells in the brain results in gliomas in nearly all mice within a few months. This technique obviates germline manipulation, which can be unpredictable and time-consuming. It also allows for the study of cell-type specific effects of particular oncogenes in vivo. Here we show a hematoxylin and eosin stain of a mouse brain tumor derived from the RCAS-TVA model. This tumor was generated in a nestin-driven TVA mouse, following inoculation with viruses bearing RCAS constructs for PDGFA, an oncogene, and short hairpin to TP53 and NF1, both tumor suppressor genes. As you can see, these tumors, demarcated in red, recapitulate some of the features found in human glioblastoma, such as an infiltrating tumor front, pseudopalisading necrosis, and intratumoral hemorrhage. These tumors have been characterized and also shown to demonstrate genetic and epigenetic similarities to human glioblastoma, making the RCAS model ideal for preclinical study of tumor initiation, progression, and response to therapy. In conclusion, genomic and epigenomic alterations are now part of the diagnostic criteria for specific types of CNS tumors. This indicates that molecular tools will become more important for not just diagnosis, but also stratification of patients who require more aggressive treatments or closer surveillance. With advances in these tools, we expect the discovery of more clinically relevant genomic and epigenomic alterations. The RCAS-TVA mouse model recapitulates human glioblastoma and represents one example of several mouse models that could uncover the functional relevance of these alterations during tumor initiation, progression, and treatment.